Chapter 4, Section 3, Cell Division, starting on page 129 of your Life Science textbook. Standard focus for this unit is that students will know that cells divide to increase their number through a process called mitosis, which results in two daughter cells with identical sets of chromosomes. Students will know that DNA, or deoxyribonucleic acid, is the genetic material of living organisms and is located in the chromosomes of each cell. Key ideas are key questions. What events take place during the three stages of the cell cycle? And how does the structure of DNA help account for the way in which DNA copies itself? Okay, page 129. In the early autumn, many local fairs run pumpkin contests. Proud oak growers enter their largest pumpkins hoping to win a prize. The pumpkin below has a mass greater than 600 kilograms. This giant pumpkin began as a structure inside a small flower. How did the pumpkin grow so big? A pumpkin grows in size by increasing both the size and number of its cells. A single cell grows and then divides, forming two cells. Then two cells grow and divide, forming four, and so on. This process of cell growth and division does not occur only in pumpkins, though. And many cells in your body, in fact, many cells in your body are dividing as you read this page. Stage 1, interphase. How do little pigs get to be big pigs? Their cells grow and divide over and over. This regular sequence of growth and division that cells undergo is called the cell cycle. During the cell cycle, a cell grows, prepares for division, and divides into two new cells, which are called daughter cells. Each of the daughter cells then begins the cell cycle again. You can see details of the cell cycle in figure 12. Figure 12. All right, so here's figure 12. We will get to that. Notice that the cell cycle is divided into three main stages: interphase, mitosis, and cytokinesis. The first stage of the cell cycle is called interphase. Interphase is the period before cell division. During interphase, key idea, during interphase, the cell grows, makes a copy of its DNA, and prepares to divide into two cells. So to repeat, during interphase, the cell grows, makes a copy of its DNA, and prepares to divide into two cells. Growing. During the first phase of interphase, first part of interphase, the cell grows to its full size and produces structures it needs. For example, the cell makes new ribosomes and produces enzymes. Copies are made of both mitochondria and chloroplasts. Copying DNA. In the next part of interphase, the cell makes an exact copy of the DNA in its nucleus in a process called replication. Recall that DNA is found in the thin strands of chromatin in the nucleus. During the interphase, a chromatin appears as a dense mass within a clearly defined nucleus. DNA holds all the information that the cell needs to carry out its functions. Replication of DNA is very important since each daughter cell must have a complete set of DNA to survive. At the end of DNA replication, the cell contains two identical sets of DNA. You will learn the details of DNA replication later in this section. Preparing for division. Once the DNA has replicated, preparation for cell division begins. The cell produces structures that it will use to divide into two new cells. At the end of interphase, the cell is ready to divide. Stage 2, mitosis. Once interphase is complete, the second stage of the cell cycle begins. Mitosis is a stage during which the cell's nucleus divides into two nuclei. Key idea here. During mitosis, one copy of DNA is distributed into each of the two daughter cells. So to repeat, during mitosis, one copy of the DNA is distributed into each of the two daughter cells. Scientists divide mitosis into four parts or phases: prophase, metaphase, anaphase, and telophase. During prophase, a thread-like chromatin in the nucleus condenses to form double rod structures called chromosomes. Each chromosome has two rods because the cell's DNA has replicated and each rod in a chromosome is an exact copy of the other. Each identical rod in the chromosome is called a chromatid. Notice in figure 11 here that the two chromatids are held together by a structure called a centromere, right there. As the cell progresses through the metaphase, anaphase, and telophase, the chromatids separate from each other and move to opposite ends of the cell. Then, two nuclear envelopes form around the new chromosomes and at the ends of the cell. Right, so here we have figure 12 that we were going to discuss. So this is the order of the cell cycle. Cells undergo an orderly sequence of events as they grow and divide. The sequence shown here is a typical of a cell cycle in an animal cell. So in stage 1, interphase, the cell grows to its mature size, makes a copy of its DNA, and prepares to divide into two cells. Two cylindrical structures called centrioles are also copied. Sorry. Then the second part, mitosis, right? The first part of that is called the prophase. Chromatin in the nucleus condenses to form two chromosomes. So we have the chromatin condensing to form chromosomes. The pairs of centrioles move to the opposite sides of the nucleus. Spindle fibers form a bridge between the ends of the cell, and the nuclear envelope begins to break down. So here's a diagram, and here's a photograph of a cell undergoing that process. And the second part of mitosis is called the metaphase. The chromosomes line up across the center of each cell. Each chromosome attaches to a spindle bar at its centromere, right? So the chromosome starts spreading apart, right? And you see an image of it taking place here. And the third part of mitosis, the anaphase, the centromere is split and the two chromatids separate, and each chromatid becomes a new chromosome. The new chromosomes move to opposite ends of the cell, and the cell stretches out as the opposite ends are pulled apart. So the centromere, which is the little like a rubber band thing that holds them together, dissolves, right? Splits apart, and these spindle fibers then contract and drag each chromatid to the opposite end of the cell. And then we move to the fourth phase of mitosis, the telophase. The chromosomes begin to stretch out and lose their rod-like appearance, and a new nuclear envelope forms around each region of the chromosomes. The DNA is once again separated from the cytoplasm by a membrane. So a membrane forms around, okay, so a nuclear envelope forms around each set, each copy. And you see an image over here with basically two nuclei are forming. And then at the end, cytokinesis. The cell membrane pinches in around the middle of the cell. The cell splits in two, and each daughter cell ends up with an identical set of chromosomes and about half the organelles. Right? Then each cell grows new organelles, it grows in size, and the process starts over. So the final stage of it is cytokinesis. Right? The final stage of the cell cycle is called cytokinesis. Cytokinesis completes the process of cell division. During cytokinesis, the cytoplasm divides. The organelles are distributed into each of the two new cells. Again, it's a key idea, so I'm going to repeat it. During cytokinesis, the cytoplasm divides. The organelles are distributed into each of the two new cells. Cytokinesis usually starts about the same time as telophase. When cytokinesis is complete, two new cells or daughter cells have formed. Each daughter cell has the same number of chromosomes as the original parent cell. At the end of cytokinesis, each cell enters interphase and the cycle begins again. Cytokinesis in animal cells. During cytokinesis in animal cells, the cell membrane squeezes together around the middle of the cell. The cytoplasm pinches into two new cells, and each daughter cell gets about half the organelles. Cytokinesis in plants. Cytokinesis is somewhat different in plant cells. The plant cell's rigid cell wall cannot squeeze together in the same way as a cell membrane can. Instead, a structure called a cell plate forms across the middle of the cell, and the cell plate gradually develops into new cell membranes between the two daughter cells. New cell walls then form around the cell membranes. Structure and replication of DNA. DNA replication ensures that each daughter cell will have the genetic information it needs to carry out its activities. Before scientists could understand how DNA replicated, they had to know its structure. In 1952, Rosalind Franklin used an X-ray method to photograph DNA molecules. Her photographs helped James Watson and Francis Crick figure out the structure of DNA in 1953. The structure of DNA. If you were to unravel a chromosome, you would find that the DNA strands are wound tightly around proteins. The proteins help support the chromosome structure. Pardon. Notice in figure 14 that the strands of the DNA's molecule look like a twisted ladder. The two sides of the DNA ladder are made up of molecules of a sugar called deoxyribose, alternating with molecules known as phosphates. Each rung is made up of a pair of molecules called nitrogen bases. Nitrogen bases are molecules that contain the element nitrogen and other elements. DNA has 4 kinds of nitrogen bases: adenine, thymine, guanine, and cytosine. The capital letters A, T, G, and C are used to represent these 4 bases. The bases on one side of the ladder pair with the bases on the other side. Adenine, A, only pairs with thymine, T, while guanine, G, only pairs with cytosine, C. This pairing pattern is the key to understanding how DNA replication occurs. And so here we see, right, thymine bonding with adenine, guanine with cytosine, cytosine with guanine, adenine with thymine, right? And the strand itself is an alternation of deoxy, which is deoxyribose, which is a sugar, and a phosphate, which is marked here in purple. Right? DNA replication begins when the two sides of the DNA molecule unwind and separate, somewhat like a zipper unzipping. As you can see in figure 15, right, here we go. The molecule separates between the paired nitrogen bases. Next, nitrogen bases that are floating in the nucleus pair up with the bases of each half of the DNA molecule. Key idea, because the way in which the nitrogen bases pair with one another, the order of the bases in each new DNA molecule exactly matches the order of the original DNA molecule. So to repeat, because of the way in which the nitrogen bases pair with one another, the order of the bases in each new DNA molecule exactly matches the order in the original DNA. Adenine always pairs with thymine, while guanine always pairs with cytosine. Once the new bases are attached, 2 new DNA molecules are formed.